everyone, this is Gayatri Prem Kumar over here, and today we are going to discuss about one of the minor experiment that is about study of plant population. Under this itself you have two minor experiments, that is plant population density and the other one is the plant population frequency. And the examination point in these two minor experiments, you will be getting any one, either the plant population density or the plant population frequency, and that carries 4 marks. And in this video we are going to discuss about the basic introduction, like what is population density, what is population frequency. And the reason why we are discussing these introduction is that the viva questions, so viva questions will be asked based on these basic concepts, so you should have an idea about what is population definition, density, population frequency. So considering this, we as usual, like all the experiments, we do have aim, requirements, and then the procedure, observation, and here we do have a tabular column, because we just make a tabular column based upon the data what we study, and we make a tabular column, and following that what you observe, and finally you write the end result and the precautions that has to be followed when you are doing this plant population density or frequency, that experiment. Okay, now coming back to the inner content. So what is the basic introduction? What is the basic definition for the population? It is nothing but a group of organisms present in an area. And generally we all know that population, whatever be the reason, so the population number, the number of organisms present in an area may keep increasing or decreasing. Now what is population density? Definition for population density, how do you find out? So basic formula is to find out the population density is number of individuals present in all the quadrat. So I'll also explain you what is quadrat. So number of individuals present in all the quadrat divided by number of quadrat you study. So what is quadrat is, like you take a sample area. So in the garden, for example, the garden, sometimes the plant population will be more dense or it will be very less. So you should know how to fix the size of the quadrat, whether you take a 10 into 10 centimeter or you take a 20 to 20, 50 to 50, 60 to 60 whatsoever. To fix the quadrat, you have to start taking 10 centimeter. Quadrat is the size what you are studying. Randomly you take like 5 places, randomly you take 5 quadrats, and this density is number of individuals totally. Suppose you take 5 areas, 5 quadrats you make. Quadrat means is that nothing but simply the area what you are going to measure. So how much area you are going to measure? It could be either 50 centimeter or 60 centimeter, but the same size should be there for all the quadrats. Okay, now what is population density? How many individuals totally you make? Like this, you make different quadrats, different samplings you make it, no? So how many individuals are present in all the quadrat, that divided by number of quadrats you studied. And in each sample area I have the number of organisms per plant population, so that I make it as a total number. That will be the population density. Again, coming back to the population frequency, total number of quadrats in which species occurred. Sometimes what happens, some quadrats will have some organisms, in some quadrats there will not be any organism. So number of quadrats in which species occurs means what? That particular organism is present by total number of quadrats you are studying. Suppose I study 7 or 8 quadrats, I find out different sample areas 7 or 8, but I can find organisms only in 5 quadrat. So what I will be writing? 5 divided by 8. So this 5 stands for number of quadrats in which species occurred, divided by I am studying 8 quadrats. So 5 by 8 into 100, that will give me the population frequency. Here for example, number of individuals in all the quadrat. So for example, when I count the number of organisms in each quadrat, like 5 in 1, 7 in 1, 8 in 1, together suppose if I am getting 37 individuals in all the quadrat, divided by S stands for number of quadrats that you study. So I am studying suppose 7 quadrat. So divided by that will give me the population density. Anyhow, we'll discuss about that in detail in the tabulation area. When I'm doing the tabulation, that time I'll tell you in detail. So this is an overall idea about what is population density and population frequency. And now we'll discuss about the aim, requirement, procedure and other details. For example, like you take, for example, I'm making a rough diagram. So you make for 10 centimeter, 20 centimeter, 30 centimeter, 40 centimeter, like that. You keep making it for 1 meter, and you find out how you make this, like using a string. So first you plot it with the nail, and you find out for every 10 centimeter what is the organisms present of that particular area. What you will do is you will randomly fix any 5 or 6 or 7 quadrat based on the minimum size. For example, I have taken here as 40 centimeter, so that they will decide it as the size of the quadrat. Then what I am doing is, so I just I have taken just 5 plant species. Randomly I have taken 5 plant species. So, and I have given them as A, B, C, D and E. And then for each plant species I have given one pictorial representation also. And now, for example, in the quadrat 1 of that 40 centimeter, I have these kind of different plant population in the quadrat 2, 3, 4 and 5 respectively. Now I am going to find out the data. I am going to fill in the tabular column of what the plant species I am getting in each quadrat. For example, this is plant species A. So what is the plant species A? How does it looks like? It looks in this shape, for example. Now what is the number of that plant species in each quadrat? I have to count. 1, 2, 3. So I write that number here. Again, if you see in quadrat 2, it is 1, 2, 3. So that number I am writing. Likewise, I am filling the data of each plant species present in each quadrat in the tabular column. The next thing what I am doing, I am doing it for the plant species B. And then what is the plant species B shape? Then for the plant species C. And then for the plant species D. And finally for the plant species E. So these data I am filling present in each quadrat. Now just check and tell whether all the quadrat contain all the plant species. No. If you see, the plant species E is not present in the third quadrat. Okay. Now this data I have got. Now I have to find out what is the total number of individuals in all the quadrat. I count that and write the data for all the 5 quadrats. Next, what I am going to do, I am going to find out what is the total number of quadrats in which species occurred. So if you see here in all the 5, the species, all the 5 quadrats, the species is present, but here only 4 quadrats, the species is present. Next, what you will do, how many quadrats you have studied? You have studied for 5 quadrats, so that data you will fill in. Now comes the important part for population density and frequency. In your minor experiment, you will get either population density or frequency, not both. So if you are getting population density, you have to write all the other things as it is. But you will be writing the calculation, total number of individuals in all the quadrats. So for example, it is 12, no? So 12 divided by what is the total number of quadrats studied? 5. So you just divide it. When you divide it, whatever the answer you are getting, so that you will write it as the population density of that particular species in that particular species A by adding all the 5 quadrats. Then you do it for B, C, G and E. This is how you will do the population density. In case of population frequency, you will not do this part. You will do only the frequency part. So what you will do, total number of quadrats in which species occurred. Here the species occurred in 5. Divided by total number of quadrats studied is 5. So 5 by 5 into 100 is again 100%. But here you will not get 5. It is only 4. So 4 by 5 into 100%. You make that and find out the calculation. This is what you have to do for the population frequency. And finally, you have to conclude your result by saying that whichever plant species is more. So this plant population density is more in that particular area, or this plant species is less in that particular area, and this plant species is more frequent or less frequent. So thereby you can give your conclusion, or thereby you can give your ending result or observation based on the plant species. And what are the precautions that has to be followed when you're doing this experiment? So coming back to the precautions, the string what you're using for measuring the quadrat should be thin enough, and then you have to be very accurate in finding out the sample. Like for example, the quadrat size means you should not take into account the organisms or plant species which are present outside that quadrat. Only inside should be taken into consideration. And what is the application of this application of this study of plant population is that you have the chapter organisms and population in your theory, where you study about the mortality, natality, and how to find out the population size, and then the carrying capacity. All these things you relate with the organisms and population of that particular area. Keeping this into mind, how to sample an area and how the organisms keep changing and how to find out the population density. So this would be the application of this practicals just for the laboratory purpose and just for the knowledge of understanding, because we cannot go and make a census of a big area or big forest. To do it in that way, this is small application, you're doing it in the laboratory level for your biology board practicals. Hope you understood what is the basis of this study of plant population, and this is a minor experiment carrying 4 mark. In this plant population, you will be getting either population density or population frequency, not both, because any one only you will be getting as a minor experiment. And this is the way, and you have separate mark for the tabulation and precaution, because this is the only experiment where you have the tabular column, and however the data is given to you, you have to represent the data and write it in the tabular column. And as usual, you have the aim, materials required, procedure, and other things should be filled in with the precautions. That is very important. And this is also related to the viva. When viva questions is asked, you should know what is population density and other definitions which is relating with the topic. Hope you understood this video and had some information. Thank you.